Welcome today, Pratibha Singh. Pratibha is, like she mentioned, she's doing her study or research in Lund University. And she's working with Isabel Gonzalez and Andreas Etzfeldt in Lund. And today she's going to present her recent research about the role of MMP2-dependent TGF-B2 activation and how it impairs fibrous cap formation in type 2 diabetes. So like Pratibha just mentioned, her focus is very much on diabetes associated atherosclerosis. So it's wonderful to have you today, Pratibha. Please tell us more about your research.
Thank you. Thank you for your kind information. Introduction. So yes, I am here to talk about one of our work which has been recently published in Nature Communication. And in this work, we have tried to explore the role of fibrous cap in providing the stability on the atherosclerotic plaques associated with the type 2 diabetes patient. Uh, Okay, so this is the paper, and it has been quite a journey, and there are many members of our group which has been involved and has contributed very significantly to this publication. And we were also grateful to our collaborators and their feedbacks, positive feedback, making us possible to publish this paper. And today I will walk you through the key findings of this paper, what it means, and what we have understood from the results that we have got it with our research. So to give you a slight context, before we dive into the results, I'm sure everyone knows what atherosclerosis is. It is a condition of the arteries which is characterized by buildup of plaque in the inner lining of these arteries. And it is the rupture of these plaques which causes the clinical complications or the cardiovascular events such as myocardial infarction or the stroke. And the stability of these plaques are quite dependent on the fibrous cap. And when this fibrous cap, the main function of these fibrous cap is to provide the integrity or the stability to the plaque from the lipid-rich core to get exposed to the circulating blood. So when this cap is weakened or the plaque ruptures, it triggers the clot formation, and which is the major reason for the clinical events that followed the ruptures of the plaques. That's why they are the people with the vulnerable plaque are at higher risk of experiencing the cardiovascular event. So, but in this publication, we were mostly interested in studying the atherosclerosis that is associated only in the type 2 diabetes patients. And I will tell you why, because these patients, they experience an aggressive form of atherosclerosis. There is an increased formation of the plaque, as well as they have a higher frequency of plaque ruptures. And despite of these patients getting the treatment for their major risk factors, they still are at much higher elevated risk of having the cardiovascular event in their life. So, and inconsistent with this, several imaging studies have also shown that the plaque of the type 2 diabetes patients are characterized as a thin cap fibroatheroma. So they have a really thin fibrous cap, which further suggests that they have the vulnerable plaque phenotypes. So all these studies actually highlights that we are still not fully addressing the underlying biological mechanism, which is contributing to the risk of plaque ruptures in type 2 diabetes patients. So in this context, in our group, we have done an earlier study in which we compared the plaque stabilizing as well as destabilizing factors. And what we have seen through this study is that although there was not much difference in the inflammatory component or the destabilizing component between the two groups. However, the type 2 diabetes groups are notably much more deficient in the plaque stabilizing component, which kind of contributes to the higher risk of plaque ruptures in these patients. So, in an effort to understand the impaired fibrous repair process in type 2 diabetes subjects, we have continued with our current study. So, the plaque samples, the carotid plaque sample, the patient blood, follow-up information about the patient, and the plaque homogenates and all other resources that we have utilized in this study is actually obtained from CP Biobank. And this biobank is established by Professor Isabel Goncalves and Associate Professor Andreas Esfeld. So we use all these resources for different assays to investigate the plaque characteristics. To start with the results, so the first thing what we have done is we have confirmed from like in our CPIP cohort, just like the imaging study, that type 2 diabetes plaques, they have very smaller fibrous gap. And not only this, when we looked at the histological component, which can impact the plaque vulnerability in these type 2 diabetes patient, we have find out that type 2 diabetes patient with highly vulnerable plaques are the one which has higher risk of post-operative cardiovascular event. And when we looked at individually at the component which has contributed to this risk of cardiovascular event, the two histological component that are quite related to this high risk are the smooth muscle cells and the collagen levels. And we know that the collagen from the previous studies, we know that collagen is produced by the growth factors. So the first thing that we did is we measured several growth factors into the plaque. And we analyzed these growth factors to understand which of these could be of importance and play a key role in separating the type 2 diabetes plaque from the non-diabetes plaque. So from this analysis, it has been revealed that the 3 TGF-beta isoform are the key growth factors that are involved in separating type 2 diabetes plaque from the non-diabetic plaque. And notably, what we have also seen that all the three TGF-beta isoforms has been reduced in the type 2 diabetes plaques. In addition to this, we have also found that the cultured cells of the plaques from type 2 diabetes patient releases much more lower amount of the 3 TGF-beta isoforms into the culture media. To understand further, like which cells of these plaques are actually causing the release of the TGF-beta2, we have taken help from we analyzed the single cell data sets. And what we have found from this analysis is that TGF-beta1 is expressed mainly by the CD45 positive cells, whereas all three TGF isoforms were expressed by the non-immune cells, which are the CD45 negative cells. And then we have seen the similar expression of all three TGF-beta isoforms from the non-immune cells in the publicly available data sets that we have used as a validation cohort of our findings. So then, knowing that it is the non-immune cells that are the major producers of TGF-beta2 in the plaques, we wanted to understand which non-immune cell specifically are the main source of the TGF-beta2. And for that one, we have done the clustering and we discovered the 4 clusters of the vascular smooth muscle cells, which we have annotated as a contractile vascular smooth muscle cell, adipocytes, like vascular smooth muscle cells, synthetic or fibroblast like vascular smooth muscle cell, and endothelial and macrophage like vascular smooth muscle cell. We also find a cluster of the endothelial cells in our CD45 negative plaque cells population. And these clusters were annotated based on the DEG as well as the cell type specific marker and expression of transcription factors. And by looking at these clusters, the expression of 3 TGF isoform in these clusters, what we have understood is that it is the TGF-beta 2 which is expressed highly by the contractile clusters, the cluster 1 compared to all other clusters. And we confirmed this thing in vitro also, where we have compared the 3 TGF-beta isoform gene expression in synthetic versus contractile vascular smooth muscle cells. And we do see that there is increase in the TGF-beta 2 by the contractile vascular smooth muscle cells. So we wanted to understand of all the 4 vascular smooth muscle cells that we have discovered or found in our non-human cell type, which one is actively involved in the repair processes. And for this reason, we did the pathway analysis. And what we have seen is that it is the cluster 3, which is involved in the active repair processes, because this cluster is enriched in several terms related to extracellular matrix organization or the collagen formation of biosynthesis. So when looking at the genes that are involved in these pathways, we have found out that several collagen genes were expressed by the cluster 3 compared to all other clusters of vascular smooth muscle cells, kind of suggesting that this is the cluster that is actively involved in repair process in the plaque. But what we have not shown yet is if these TGF-beta isoforms are related to the collagen. So we did another association analysis, and via a hierarchical analysis, we have seen that the collagen is closely associated with all 3 TGF-beta isoform than all other growth factors that we have measured in the plaque. And interestingly, what we have found out that TGF-beta 2 show a very strong positive correlation with the collagen in type 2 diabetes plaques. So I wanted to further validate this connection between collagen and TGF-beta 2 in vitro. So we did in vitro experiments. And in these experiments, what we have done is that we have stimulated the smooth muscle cells synthetic in their proliferative state with the increasing doses of 3 different TGF-beta isoform. And we confirmed that these cells are stimulated by these 3 TGF-beta isoform by looking at the phosphorylation level of the SMART2. And we see that there is an increase in the phosphorylation level of the SMART2. And what this stimulation does is actually it increases the expression of collagen gene from these cells. So to mimic the in vivo setup, where we think that the crosstalk between the synthetic cell and the contractile cells are actually important for the release of collagen from the synthetic cells, we have done an alternate experiment. And in this experimental setup, what we have done is we have stimulated or incubated the synthetic smooth muscle cells with the media derived from the contractile cells that has been cultured for 6 days. And what we see that after 72 hours of incubation of the media derived from the contractile cell is still able to increase the collagen gene expression from the synthetic cells. So we see the very similar finding in both the experimental approaches. So far, what we have seen with our result is that TGF-beta 2 is important for the factor for the plaque stability, and it is produced by the contractile vascular smooth muscle cells of the plaque, and it enhanced the vascular repair processes by acting on synthetic vascular smooth muscle cell and influencing the collagen production from these cells. But what we have not explained yet is that what causes the decrease of TGF-beta2 in type 2 diabetes plaque. So then we thought of two possible mechanisms that could account for this decrease. And one of the mechanism is possible. There is a possible regulation acting at the level of TGF-beta2 cleavage and release from the contractile vascular muscle cell, or there could be an overall decrease in the number of TGF-beta2 producing cells in these patients. So the next set of experiments were done to understand these two possible mechanisms, what could cause the TGF-beta to decrease in type 2 diabetes patient. So looking at the regulators of TGF-beta to cleavage, the earlier studies have demonstrated that MMP2 could be important for cleavage of TGF-beta2 and activation of its downstream signaling. So we looked at the MMP level, like several MMP levels in the plaque. And out of various MMP levels, it's only MMP2 and MMP9 that has found to be detected in enough amount to be able to do the analysis. We see that MMP2 level is decreased in type 2 diabetes patient, and it is produced by the synthetic cells of the plaque. And when we correlated the MMP2 and 9 levels with the different TGF-beta isoforms, we have seen that MMP2 level correlated positively with the non-diabetes plaque with the TGF-beta 2 levels. However, the MMP levels did not correlate with the TGF-beta levels in the type 2 diabetes patient, which kind of suggests us that there might be a disruption of the MMP2 activity in type 2 diabetes plaques. So we next explore if there is what happened to the activity of MMP2 in type 2 diabetes. We do see that the MMP2 activities were decreased in type 2 diabetes plaque cells. And to understand what could actually cause this decrease of the MMP2 activity, we did the in vitro investigations again, and we found out that it is the hyperglycemia which has suppressed significantly the MMP2 activity of these cells. So then next we wanted to explore our second question, which is if there is a decrease in the proportion of the cells that express the TGF-beta2 into the plaque. And for this analysis, we did the spatial transcriptomics. And now with the spatial transcriptomic, we have mapped all our 4 clusters into on top of the plaque. And what we have seen from this spatial transcriptomic is that the contractile cell cluster is present into the plaque cap region, which go localized with the TGF-beta2 positive spots. And there's also some, and this region is heavily populated by the contractile cells, and there is only few synthetic smooth muscle cells present in this region. The synthetic smooth muscle cells were actually found more into the media to intima to media interface region. So, and also we further compared like the proportion of the TGF-beta to positive and negative spots between type 2 diabetes and non-diabetes patient. And what we have seen that the type 2 diabetes patient has a much fewer TGF-beta 2 spot compared to non-diabetes patient, which again validate our previous finding that TGF-beta 2 is much less in type 2 diabetes plaques. And when we look at the clusters of interest, we have seen, and a very interesting thing is that the contractile cell clusters is quite, the number of contractile clusters were quite low in type 2 diabetes plaque, whereas the number of the spots predicted as synthetic clusters are quite high in type 2 diabetes plaque. So we think out what phenomena, how could we explain this finding, and we thought of there could be an impairment of the fibrous repair process happening in the cap region of the plaque. Because this, we think that the TGF-beta2 is less, probably due to the less amount of the contractile cells. The synthetic cell did not are not able to change back to the contractile phenotype cell type within the cap region. So to confirm this one, we did another in vitro experiments where we have incubated the synthetic smooth muscle cells with TGF-beta2, and we have seen that the TGF-beta2 actually is helping these cells to differentiate into the contractile phenotype. And this was confirmed by increased expression of all the contractile phenotype cell markers. To summarize this, this is the summary. And what we have seen in our study is there are two different cell types, and TGF-beta2 is the main factor regulating the plaque stability. And it does that by stimulating the synthetic smooth muscle cells for active fibrous repair process through production of ECM or the collagen production. So what happened in type 2 diabetes is that the activity of MMP2 is impaired due to hyperglycemia, and because of this impaired MMP2 activity and also the level, there is not enough release of TGF-beta2 from the contractile vascular smooth muscle cell. And this shortage of TGF-beta2 then again affect the synthetic vascular smooth muscle cell to differentiate back into the contractile vascular smooth muscle cell, creating a kind of negative feedback loop, resulting in the impaired fibrous repair process and thus causing destabilization of plaque in type 2 diabetes patient. So yes, this is all about my paper, but there are also some key review points that I would like to bring up. And these points has actually help us. It's quite valuable feedback from the reviewer and help us improved our paper. And one of the concerns that has been raised by one of the reviewer is about the possible contamination of plaque cell preparation with the blood cells or the circulating cells, because the reviewers has found that these monocytes were quite abundant in CD45 positive cell types that we have shown in our older manuscripts. So in that response of the reviewer concern, we have said that there are several steps that we have taken to avoid blood cell contamination at plaque cell preparation level, and that includes the thorough washing of the plaque with the media and using the red blood lysis buffer before we mince the plaque or start the enzymatic digestion of the plaque. And at the level of sorting of the blood, like these cells, also we have another step to avoid the contamination by the blood cells. So we gated out all the RBCs that are stained with CD235 positive, and we sorted the CD45 positive and negative cells after gating out cells they are stained for CD235. And then there is also, and I would just show you that to support our point, we have given the evidences, so we have given the cascading strategies, and you can see when we sorted the CD45 positive cells, they are the RBCs are gated out, and we have a population of CD45 negative cells, and even in the CD45 positive cells, after the sequencing, we have checked the gene expression of two erythrocyte markers, and we did not see any erythrocyte contamination in the CD45 sorted cells. So it's highly unlikely that we have the blood cell contamination in our blood cell preparation. And reviewers concerned, I mean, we could not completely exclude the presence of blood-derived monocyte in the plaque, but to this end also, we have changed our reference, which we used before to predict the cell type. So because we were using I will show you that we are using the blueprint and ENCODE as our reference data set to annotate, to predict the cell type. And this reference data set consists of RNA-seq samples from stroma and immune cells. So it is very important to use the correct data set, which resembles, for the cell type prediction, which resemble your tissue type. So we have changed the reference data set to tabular vasculature. And this data set is used for cell type annotations in collection of cardiovascular single-cell genomic data sets found at plaqueview.com. And to further support our point that we don't think that there is a contamination of circulating blood cells, and our main focus was on the TGF to find out the TGF-beta source, isoform source into the blood cells, and we have only seen that all three TGF-beta isoforms were expressed by the non-immune cells and not the immune cells. So that kind of gives us an edge because we are not talking much about the immune cells in our current publication. And then there's also one concern from one of the reviewer is about the annotation of the vascular smooth muscle cells, that we have annotated these cells as macrophage-like or synthetic fibroblast-like, but we should confirm the identity of these cell types by showing the absence of prototypical cell markers for cell types like macrophages, fibroblast, or the chondrocytes. So we took the cell specific markers of the macrophages and checked their expression, and we did not find any significant overlap of these marker expression on the vascular smooth muscle data or the phenotype that we have annotated in our vascular smooth muscle cell. And the third reviewer feedback, which was quite useful for us, is that the reviewer has asked us that we can use the publicly available data set as our major advantage as a validation cohort. So we have used the LSI data set, and we have seen that the four vascular smooth pore clusters, just like ours, also in the publicly available data set of carotid plaque. And the gene, the DEG, is specific for our four clusters, has been found to be expressed in these clusters. And also to confirm the phenotype of our vascular smooth muscle cell cluster, we use two different approaches. So first, we have just look at the DEG specific for our cluster. For example, MYH1 is specific for our contractile vascular smooth muscle cell, and we have found that it has been expressed in this data set. ABCC9 is for adipocyte-like, likewise LUMP for fibroblast, synthetic, and CCMD1 for macrophage-like vascular smooth muscle cells. So these are expressed in the clusters. Plus, we use another approach where we have mapped our clusters over this data set, LSI data set, and we confirm that all the four vascular smooth muscle cell phenotype that we have discovered in our data set is also present in the atherosclerotic plaque core or the proximal adjacent region. So yes, this is all about this paper, and I'm very thankful to my team and everyone who has contributed to this study. I'm thankful to all the patient who has consented to participate in this study, all the support that we get from the different funding strategies. And yes, I'm very happy to take any questions.
Thank you so much, Pratibha, for this exciting work. We're happy to take questions now. Feel free to type in a question in the chat or just unmute yourself and ask a question.
Great presentation. If I can make a comment while
Yeah,
we see if
thank
there are
you.
any other
We'll
questions
warm
for the
up.
audience. Fantastic amount of work, super impressive, and so many questions. Let me try to prioritize them. Yeah, super exciting work. I was curious about, you were just talking about the clustering now, and
Mm.
I was wondering whether you delve a bit into cluster 4, the endothelial cell one, and
Yeah,
whether
there
you
were
have
four.
any comments on that.
I'm sorry, what is your question, really?
So the cluster 4, the endothelial cell one, whether you guys look a bit more into it and whether you have any comments about what you notice.
I mean, when we have done this study, we were, as you might have noticed, that we have used the SmartSeq2 platform for the RNA sequencing of these data. So then we have done 4 patient samples, and all of a sudden, the SciLife decided that we would change the chemistry of these cells and we would move to SS3. So then we have a limited amount of patient for this analysis, and from these clusters, we could find only one endothelial cell clusters, but we haven't done the subclustering of these endothelial clusters to understand if there is like even more granularity among these endothelial cells and what is their role actually in type 2 diabetes. So that's another aspect to study. But
Yeah, thank.
we haven't also found, because our focus of this study is mainly the TGF-beta isoform. So we haven't found that endothelial express so much of the TGF-beta isoform. So we focus mainly on the 4 vascular smooth muscle cells, who out of the 2 seems to be kind of crosstalk with each other in terms of fibrous repair process.
Fair enough. Yeah, no, definitely. And it's such a shame that you don't have the granularity to delve into the endothelial cells as
No,
well,
but
but
we
already.
what we yeah, what we are working on it. We have actually we moved to the another this SS3 platform, and then we have some more data, and we are working on it. And
Okay,
definitely,
okay.
there's it's quite promising with a single cell coming up.
Okay, very, very, very intriguing and curious to see more because I was just looking at the table of non-immune cells expressing TGF-beta isoforms, and the endothelial cells seem to be like among those, showing something for TGF-beta 1 and 2, but not 3, which would be in line with what we see in some transitioning endothelial cells that arise in a healthy condition such as atherosclerosis and diabetes, such as
Mm.
NMT-related cells, where
Yeah,
TGF-beta plays a role. So I was just
yeah,
wondering if
yeah, yeah,
something
yeah.
along the lines of.
We have
Okay, okay.
quite interesting
Fair enough, fair enough.
research line towards that as well. Like,
I can see that
yeah.
we think alike when it comes to that part. Yeah, well, looking forward to see more then. If there are any other questions for the audience. Let's see.
Well,
Um,
if we don't, if sorry, Melody, go ahead.
Yeah, I was just going to jump in if that's okay and ask about so in your study basically you show that it's actually MMP2 inactivation because it's inactivated that way TGF-B is not mature enough to have a stable fibrous cap. But I think what do you think about the and that's because of the hyperglycemia you're suggesting. Do you think hyperglycemia also has an effect on other MMPs as well? Because usually MMPs within athero they are related to instability because they do degrade a lot of the ECM matrix, which is thought to be a stabilizing effect in the fibrous cap specifically in athero. So what are your thoughts on hyperglycemia and other MMPs as well? Have you looked at that or?
I have I I think we have looked at it and we did some linear regression to understand if any of the MMP associate with type 2 diabetes, but it nothing that I had remember or caught my attention. Yes. Mhm.
I can comment on that with MMPs, with Andreas here.
Yeah.
We're
Hi, Andreas.
here. Hi.
Hello.
So there are several MMPs basically, and we've shown like the MMP9 is key for the rupture event in itself. But it doesn't really seem like they, as Pratibha said, that they're sticking out in terms of an increased activity or so. You have developed an advanced atherosclerosis. That is also to keep in mind. Advanced atherosclerosis that is well treated in terms of statins and so they're well treated as well.
Yes, it is. Also listen.
And I also know that the collagens as well that are within the plaque and the fibrous cap, there's a good type of collagen and a bad type of collagen as well. So even if there's increased MMP activity, we also have to see how much collagen is being degraded, if it's also like a healthy type of collagen or if it's more of a disease type of collagen as well. So the story is much more complicated than just MMPs as well. So that's keeps us in our jobs, I guess. Yeah. Any more questions?
Chris, I think you had a comment. Is that correct? Let's see.
Sorry, here I am.
Hello.
Sorry. Oh my goodness. No, I had a curiosity about the experiments that you did with the conditioned media. I was wondering if you ever looked at the possibility of the involvement of extracellular vesicles or exosomes. It's just a very general curiosity. If you see this effect by treating the cells with the conditioned media, if you ever looked at if there's certain cell types that secrete these factors in vesicles, large vesicles or small vesicles at some point.
I haven't really looked at it, but
no, just a general
point.
Curiosity.
No.
Yeah, no, definitely, because it would be probably interesting to know whether it's something in the secretome per se, or if it's something that is traveling to vehicles. That's
exactly
a nice point.
Okay, thank you.
So I think we had a great discussion. So unless there is someone shy that is preparing their question, I think one of the final comments we can probably make, if you agree, Mel, is about like also how important was to share the preparation or like how much work it takes to like go back into revision to make sure that everything is done thoroughly, or to take in any good advice from the reviewers. If you want to make a final comment on that, Pratibha, probably would be nice for the younger part of the audience.
I mean, there are things that we were suggested, and we thought that it would really improve our study. But there are also several limitations of the study that we have done. So we try to comment on each and every things that has Revere pointed out in our manuscripts what we think and why we have done it. And in a way, we have succeeded to kind of kind of have them agree with us. But I think it all depends upon, like, if you really wanted to go for high impact and you wanted to reach the wider audience, probably spend quite a lot of time in revising your manuscripts and working toward the revision. But if your end goal is to publish faster, probably is not a good idea, because for us it took almost more than a year to revise the paper and be able to be accepted and even to come online, because after revision, like after revision and acceptance, there is the whole process of editorial review for the Nature communication, and it takes quite a lot of time from you to do all those editorial all the editorial comments and review to improve your manuscript to be able to make it in a format that it get published. So, but like I said, it all depends upon your own
It's a balance,
interest.
probably, right? But yeah, also I think
It's your
good for the
interest.
youngsters to know that once it's accepted, it's not done and it's
The
not gonna
end.
be online the day after. It takes a
Yes,
lot of time. Yeah.
it takes a lot of time. I mean, I think more than revising our manuscript, we have spent quite a lot of time for the editorial process because they wanted to have well-detailed methodologies. Plus, there should be detail of there were several forms that we need to fill with all the methodologies, and we need to publish there's also a question regarding publicly making the data available, because it's an open access journal. So they wanted us to make our data available, publicly available, or deposit on this geo databases. But then, I mean, we also have our own restrictions, because we work on these data sets are from the living subjects, so we cannot go public. It comes under, the ethical review that we have associated with data set. It doesn't allow us to do that. So we have to kind of negotiate quite a lot on this way to be able to publish. Yes.
A lot of admin work behind the scenes.
Yes, this.
I think we have a final question from Luis, if you want
I
to
have
come in.
Is my microphone working?
Yes.
Yes.
Yes, okay. Hello, it was a great presentation. I'm not too versed in cell studies, so that's why I took a while, because I didn't want to make a silly question. But when I am looking at the paper that you here presented, I am questioning if you in your let's say first phase of experiments, where you had the cells from diabetic patients and other cells from non-diabetic patients, if you have any criteria to select the patients. For example, just an outstanding example, if they have, for example, cancer, maybe I won't choose this, or if they have another serious disease, I won't choose this patient. So did you have any sort of criteria when selecting the patients?
We did select the patient for type 2 diabetes. We did select the patient which has, the HbA1c level more than, now I think, was it 43 above, Andreas? Do you?
It's the full criteria here, so it's above 48, I think it is.
48.
Yeah.
Yes.
I got it was the golden criteria for, and now I got confused myself, but I can check it up. But it's the standard criteria for HbA1c levels. It's 48 right now, I think. Yeah, to be sure. But then you have the intermediate level between 40.
Yeah, because we
and
don't
48
want to include the one with the pre-diabetic condition, so we kind of use HbA1c also as one of the factors to take the patient that I would
Right.
give.
So, and this is because, and this is really me trying to learn because I don't know about cell studies. If, for example, when I do studies in biofluids, I usually need either a big cohort or I need to match comorbidities to increase the statistical significance. I know that in cells or in mice, for example, it's not the same, but I just want to learn a little bit about that. If there is a need to account for these comorbidities that the patients have to not dilute, let's say, the effect that you saw and the conclusions that you got from these experiments, so to say, okay, this was caused by this alteration and not for some other comorbidity that the patient had while it was still alive.
It's a good point. The thing is here, you can basically say what you do, you can't screen. If you take a bigger population, there will be diseases, known or unknown. And that will also be a group of individuals that we would like to study because this is how the individuals that suffer from the disease will look like in the end. And they may influence the disease, the atherosclerotic disease. But then you have, so if you do that in a large group, but then you have these subgroups like the single cell subgroup, the Visium subgroup. So basically, then you could screen and have more strict terms of what you include to be very sure. And if you can see the same pattern in all these different uh, cohorts, it basically means that it's unlikely, I would say. It's a very good question, and there will be other things that will affect, of course. But I think that is a way to make sure that it's probably our main aim in different groups that will not have all these criteria. So it's unlikely that it will be like a specific type of cancer or something driving this difference then. But it's as you say, it's an important thing, and there will always be things that we don't really know either as well, because yeah, not all diseases are diagnosed either. That is something we have to bear in mind as well.
Yeah, and then in our cohort, like we couldn't get all the clinical information about the patient, so we have like only some information that we know about.
And it's like they're not in a clinical material. We know we don't screen some diseases we screen for, right? But not not all. We have no evidence for the importance of that in a bigger perspective. But it's one of these things that we're always worried about, of course. So it's a good question, right? I think it's important thing to keep in mind.
I agree, and even the small, not small, but the detail about having to or wanting to have as much clinical data as possible is already a huge problem because usually there's not that much that we can do to obtain what is not readily available, right? Unless we are physicians as well, which I think we are not. But for example, and this was more or less what I was talking about, because age, for example, you have several factors that you take into consideration, and there might be. I'm just pulling out of my hair, but I assume you measured age, sex, BMI, glycemia levels, and so are those also taken into consideration? So, for example, there is not a big difference, a big variation between the lowest age and the highest age, or the lowest glycemia and the highest glycemia. And this is just because I'm not, I'm just literally trying to learn. I'm not saying if you did it right or wrong. I don't know about cell studies. I'm just trying to learn.
So I I can basically so again there will be differences. Most of the cardiovascular disease. Pratibha, maybe I'm interrupting now. Sorry.
No, no, it's okay.
It will affect the cardiovascular disease, as you point out. Age is important, sex is important, and there is some association with TGFs and sex. But again, then basically what we did to avoid this again is these smaller groups for single cell and for the Visium. where we streamline the age as well to have less spread of age and try to balance it gender-wise as well to keep it more balanced. But if you look in a larger group, just for like where you measure proteins to see in a larger set, there will be changes also that is associated with age and so on. But we try to adjust these for these factors as much as possible. And also when looking at the clinical factors, then again, that associates with the TGF levels, where we kind of draw hypothesis that hypoglycemia is important, basically. It seems like this has a large effect on the TGF-free TGF levels in the tissue.
All right. Perfect. Thank you.
Thank you so much for this wonderful comment. And thank you so much, Pratibha and Andreas, for presenting the work and then also interacting a little bit and the follow-up discussion. It's really great to have you present today. And if you're interested, I can also send you some information about the network in case you would like to be members as well. But I think this is us for today. Thank you so much for taking part of these monthly journal clubs. And I'm really looking forward to the next one. We can send you some more information about that in a couple of weeks. Thank you all for joining.
That will be really nice.
Thank you
Thanks
so
a
much for
lot.
joining. Yeah, it was very, very
Thank
useful. Very nice to have you presenting the
Thank,
work.
thank you. It's a pleasure.